ecology search. And it's a pleasure today to welcome Dr. Tadashi Fukami, who who comes to us with a distinguished career already. He received his undergraduate degree and graduate degree in Japan. His undergraduate degree at the University or at Waseda University in Tokyo, and his master's also in Tokyo at the University of Tokyo. Then he was a PhD student in Tennessee, and his advisors were Jim Drake and Dan Simberloff, who many in this audience know quite well for many years. Then he finished his PhD in 2003, and then took, interestingly, a postdoc in New Zealand with a soil ecologist, and brings a diversity of specialties to us as well. He's now been an assistant professor at the University of Hawaii, Manoa, for 2 years. And the title of his talk, which is very engaging, a lot of us have been interested in these issues for many years, it's entitled Historical Perspectives on Ecological Community Assembly. So welcome.
Okay.
Enjoy it.
I hope this wasn't too distracting. And yeah. Well, thank you, John, for a nice introduction, and hello everyone, and thank you for coming to this seminar. So as with many other disciplines, I'm sure Stanford is clearly one of the leading universities in the world in the field of ecology, and I'm really very much honored and excited to be invited here. And yesterday I had a wonderful time at Jasper Ridge Biological Preserve with Nona Cherriello, if I can pronounce her name right. And she was kind enough to spend a whole morning with me yesterday to show me around in the preserve. And I think it's great that you have such a nice field site so close to the main campus. And it's definitely something that I like to be to make use of if I come here for teaching, both for teaching and research. And I also had a great time talking to some of you already this morning, and I'm looking forward to talking to as many more of you as possible this afternoon and also evening. So as ecologists, we are interested in understanding a bunch of things at several levels of biological organization, starting from individuals, what happened to individuals, to populations, to communities, which are really sets of populations of different species living in a given location, to ecosystem, which include not only biological but also physical and chemical components of the given place that interact with the biological community. So to understand any one of these, you really often have to consider the others as well. But as a research focus, my interest has mainly been at the community level. And one of the basic questions that people we are interested in asking is what determines what we call community structure, which is really about species diversity, which is just the number of species you find in a given location living together, or things like species composition, the set of what kind of species you find coexisting in a given location. And this question about community structure turned out to be really complex, and there are a number of ideas, hypotheses, and theories that have been proposed to explain community structure. But if you were to ask an ecologist to name single most important and influential ideas in explaining community structure, I would bet that most of them, if not all of them, would pick this one, the one called Theory of Island Biogeography, proposed almost 40 years ago by now, or more than 40 years ago by now, by Robert MacArthur and E.O. Wilson. So what this theory does is to try to explain local communities on islands like this one, just as a simple result of a balance between the rate of species immigration coming from some mainland as a source of species and a rate of extinction that's happening within communities. So it's so what this theory says is it's just a dynamic balance between rate of immigration and rate of extinction. And this theory is so simple and nice that you can apply to this one to many types of organism habitats. For example, you can think about trees on these islands and think about tree species diversity and try to apply this theory. Or you can think about insects or maybe even bacteria or any other things. And it doesn't really have to be true islands. You can also apply something, any kind of discrete habitat like this ponds or lakes, anything. You can try to apply this theory to try to explain and predict species diversity particularly. So this theory is nice and simple so that you can apply to any kind of habitats and organism, but that simplicity and generality comes with a cost, of course, and there's a lot of variation in species diversity that remain to be unexplained if you just use the theory, and there's a lot of effort going on since this MacArthur and Wilson to try to kind of build on this theory to come up with a better understanding of species diversity. One of the, to me, the really important thing is to think about not only just immigration rate, but also immigration history to try to explain the species diversity in other kinds of community structure. So let's, to explain what I mean by this, let's suppose that we have multiple local communities in a given location, and that can be islands like this, or again, it can be aquatic ponds or lakes, any kind of discrete habitats. And if you have this, let's also suppose that these local communities share identical environmental conditions, just for the sake of argument. And let's also suppose, as a hypothetical simple example, that we have a species pool consisting of just 6 species, and one of them is carnivore, one of them is herbivore, and 4 producers. And what these work that I cite here, and many other work, what they have been saying, at least from the theoretical point of view, is that even having the same kind of environmental conditions shared by these local communities and same kind of species in the species pool, just by varying the immigration history that you use to construct these communities, you can end up having very different local community structure. So let's say, well, this is really a theoretical, hypothetical example, but let's say we have a producer introduced first or colonized in this community first, and the herbivore second in this community, and so on. If you have this particular sequence, immigration history, then you may end up having all species coexisting, but just by varying this immigration history, you can end up having different community structure. And that's been what, at least theoretically, have been proposed as a possibility. So we know that at some cases, under some conditions, it seems like immigration history has some effect on community structure. So it's important to understand, you really have to know what immigration history is in trying to understand community structure. But in other cases, maybe it's not that important to think about immigration history, and we don't really have to worry about that to understand community structure. So some cases immigration history matters, and some cases not. And it's at least when I was doing my doctoral research, my PhD days, it wasn't really clear under what particular conditions this immigration history becomes important to community structure. And that's really the main question that I have been trying to answer starting from my PhD research. So the main question I wanted to ask is this: does the effect of immigration history depend on context, ecological context? And as an example of ecological context, I'm going to go through several. One is what I'm going to call productivity, habitat size, and another one is size of species pool and habitat connectivity. All of these factors have been considered more or less kind of separately from immigration history as a really important factor determining community structure. But how this may interact with immigration history, that's something much less clear, and that's something that I wanted to look at in detail. So for the first half of my talk today, I'd like to go through each of these one by one quickly to give you an overview of what I found. So let me start with productivity. So productivity, or in this case defined as the amount of energy and nutrients available for community development, that kind of defined as that. Productivity has been considered a main determinant of species diversity. And if you have this kind of variation, so I'm using different color here to represent productivity. So if you plot productivity as your independent variable, and if you plot species diversity as response variable, what this review paper and other papers have found is that this pattern is really the common one. So as you increase productivity, you tend to see increase in species diversity, but as you increase productivity too much, then species start to decline. Whatever reason, this seem to be a common pattern we see in nature in different systems. But this is not the only pattern that we see in different natural communities. And sometimes, depending on where you look at, sometimes this could be just a monotonic increase of species diversity with productivity. In some cases, it can be negative. In some cases, U-shaped. In some cases, non-significant. And any of these, actually, it's a really messy picture here. Any of these doesn't seem to predominate over others. So we have a really messy picture here. And there has been a lot of studies trying to understand what causes this variation in productivity-diversity relationship. So in this context, what I wanted to ask is this, basic one, in relation, going back to immigration history, I wanted to ask, can immigration history be a factor in here to create variation in productivity-diversity relationship? To address that question, I could use any kind of communities, and I wanted to use something really attractive and charismatic. But it's really a problem here, because if you are interested in community assembly, you really have to look at long-term community dynamics, and you also have to be able to rigorously experimentally manipulate immigration history to be able to test this kind of hypothesis. So that's my excuse to use this microbial system. And so what I did was I had these protists feeding on bacteria, and some of them are predators of others. But in any case, I have 18 species of these species kept in the lab, and these little cups are my microcosms, model systems. And what I did was experimental manipulation of immigration histories. I had 4 different immigration histories experimentally manipulated, and I also had 5 productivity levels, because I was interested in looking at productivity-diversity relationship, and for each treatment, I had 5 replicates. So I assembled these microbial communities, and it took about a few weeks. And a few weeks is nothing for us, but if you think about these microbes, they have a generation time of roughly just 1 day. So 20 days is a really long time. So we are doing really long-term community dynamics. And after about 20 generations, after I introduced the last species, I looked at how they were doing in terms of productivity-diversity relationship. And if I use one of the 4 histories that I used, I'm going to call it A. So all of these communities are assembled by immigration history A. Then the relationship looked like this. And if you do statistics, this is a little bit of tapering off here, but more or less, it's broadly speaking, it's a monotonic increase of species diversity with productivity. Now, if I use a second immigration history, B, then this is what I got. If you try to fit any model, I couldn't, and this is really a non-significant relationship between productivity and diversity. If I use a third one, this is C here. Now this is a hump-shaped relationship that you might find in natural communities. And the last one, I have U-shaped relationship. So looking at this, it's only 4 histories, but looking at this, at least this small microcosm communities are telling us that it is indeed possible that hypothesis that I wanted to test, that the productivity-diversity relationship depend on immigration history, that hypothesis seems to have been supported by my data. But I also wanted to know what was going on. So instead of just sticking at the community-level phenomena, I went down, 1 step down, to see how each population was doing. So what I'm going to do is to show you how one of the species I had was doing and how this species was reacting to my manipulation of immigration history and productivity. So what I did was I looked at abundance of this species, particular species, and see what they were doing under different histories. And under the one of the five, this is the highest productivity one, immigration history did have a significant effect on abundance of this species. So I put significant here for now. If I go to the second productivity level, this is what I found. So it's significant response of the species to immigration history, but compared to this first one, the way history was affecting the abundance of this species were different at this level. So I'm using different color to just note that difference. If I go down to the next level, this was non-significant, and it looks like it's different, but actually statistics will tell you that this is non-significant. So I put NS here again to say that this is non-significant effect of history. I go down to the lowest level of productivity. This became significant again, but again, how the history was affecting this was different from how they were doing here. So I'm using different color here. It's getting already a little bit complicated, but just to summarize what I just showed you, for this species, sometime productivity was under some productivity levels, history was affecting this species significantly, but sometimes not. So this is just one of the species I used. And if I did the same analysis for all of the species that were surviving, at least at some of the microcosm at the end of the experiment, this is what I found. So for each species, sometimes history was affecting abundance of these species, but it depended on productivity. And maybe I haven't tried enough, but I kind of gave up in saying that it's pretty clear that I can't get a neat summary of this, but it's pretty clear at least that the magnitude and direction of a historical effect depended in a very complex way on both productivity and species identity. So looking at this productivity and immigration history, at least from my microcosm experiment results, it's saying that these two things can interact potentially to create a variety of patterns between productivity and species diversity relationship. So that was about productivity. And moving next to habitat size. Another way that these local communities can vary is in size. So sometimes you may have maybe small islands, sometimes large islands. And there's a lot of interest in ecology to try to look at how habitat size explain diversity. And this kind of increase, usually on log-log scale, has been observed very in a general way that it's been considered one of the few laws in ecology, which is a really rare thing. But if you look at data more carefully, you do find some variations. Sometimes the slope is variable depending on which communities you look at, which organism you look at, and sometimes it can be non-significant too. So in this case too, like productivity, we have some variation that we want to really explain what's going on. So similar in design to the experiment that I just showed you, I was using these microbes as model system to use for different immigration history, really similar. The only difference is that now I have, instead of productivity, I have variation in habitat size. And then I had 5 replicates per treatment. So going through 4 immigration histories I used, this one was non-significant after a long time passed after the last introduction of species. This one was significant. This one wasn't significant, and this was, there's not very neatly, but there's statistically significant relationship, positive relationship between these two. So it was a really quick summary, but just like productivity between habitat size and immigration history, there can be interactive effect between these two that can result in variety of patterns between habitat size and species diversity. Now I'd like to move on to size of species pool. So another thing we can look at in this picture, going back to this picture again, to ask whether the number of species you have in the species pool, let's say you have just a small number of species, or sometimes medium number of species, large number of species. If you have this kind of variation in the number of species pool, how that might affect how many species would be coexisting in here. And that's another question that ecologists have been interested in for a very long time, but not really so much in relation to the effect of immigration history. So what I did here is computer simulation, mathematical computer simulation using what's called Lotka-Volterra equation, which is really commonly used. One of the most basic equations that you can use if you want to simulate population dynamics that involve interactions like competition and predation between different species. Because this was getting a little bit complicated, I resort to more of a even more conceptual method here than microcosm experiments. And because I was interested in effect of immigration history in this case again, I was manipulating immigration history in this simulation to see what would be happening. So this is some of the results that I got. If you look at how the number of species in the species pool was affecting the number of species in each of these communities, if you get the mean of the number of species coexisting here, this is the pattern that I got. As you increase the number of species in species pool, you initially have an increase in number of species that can coexist in these communities. But eventually, it seems like there's some limit to the number of species you can pack into each of these local communities. And that makes kind of sense because I was assuming that there are competition and predation, those kind of negative species interactions that should limit somewhat the number of species that can coexist in each of these. But what was more interesting to me was if you assume that these local communities are not independent, but connected with one another by dispersal, occasional dispersal here, and many ecologists are now call this kind of situation meta-community. And if you look at how many species in total are coexisting in this larger spatial scale community, then I found a little bit different pattern here. If I look at the meta-community scale, the species richness doesn't seem like there's a limit, and it's more or less, it's broadly speaking, continuous increase in species richness with the number of species in species pool. So I thought this was a little bit interesting. So how this kind of scale-dependent pattern might have arisen, and that's the question I wanted to ask here, and the immigration history becomes a key here as I show you in a minute. So to see what was going on, what I did was I looked at how the community was changing over time. So for example, at this level of species pool size, this was what was happening. So over time, the communities didn't seem like they were going towards some fixed species composition, but instead, there's a continuous turnover of species over time. And given this, the next thing I looked at is to see how, if you try to introduce different species into these communities, how many species you can potentially introduce and allow these species to successfully colonize and join the community, and to look at mean number of potential successful invaders. And this is what I found. As you increase the number of species in species pool, initially there's these are actually a fixed species composition, but as you increase number of species in species pool further, the larger your species pool, the larger number of species you can use to invade these communities. So what this really means is that as you increase the number of species pool, it becomes more and more difficult to predict which species will be able to colonize these species. So as a result of that, if you look at variation among local communities, they become more and more variable, and that's probably because we see this large gap. So this is really showing that immigration history has a greater effect on local species composition under a larger species pool. So to summarize this part, size of species pool can also interact, at least from this only computer simulation, but at least theoretically, it seems like these two things also can interact through effect of competition predation to produce scale-dependent patterns between the size of species pool and species diversity in communities. Last thing I want to briefly talk about is habitat connectivity. So going back to this picture, another thing that you can vary in this picture is how connected these local communities are to species pool. Sometimes these local communities may be kind of distantly located from maybe mainland, if you think of these islands, or they can be more closely located to species pool. And that would influence how connected these are to species pool. And I'm going to call it external dispersal. So the location of these communities relative to species pool is going to affect the rate of external dispersal, how frequently species are coming into these communities from species pool. Another thing you can think about is how separately located these local communities are to one another, and I'm going to call it internal dispersal. So this is going to affect habitat connectivity as well. And using some additional computer simulation, what I found is that these two kinds of dispersal, although they haven't been really considered in combination with each other in community ecology research usually, these two rate of dispersal can actually interact through effect of immigration history to lead to any kind of this positive, negative, insignificant effect of dispersal on diversity. So it's getting a little complicated, but at least as a general message, at least what I can tell you is all of these things that I focused on as ecological context can interact with immigration history to affect community structure. So going back to this question, I would say the answer, at least from this theoretical exercise using microcosm experiments and computer simulation, the answer is overwhelmingly yes. So this is the kind of framework that I was trying to develop through my PhD research mainly. And for the rest of my talk today, I'd like to talk to you a little bit about what I have been doing since then. And what I've been trying to do is to try to expand what I call this historical perspective, looking at effect of immigration history, to expand this to have a better understanding what's going on in communities as well as ecosystem at the next level of organization. So I like to talk to you about 4 directions I've been taking more recently, and I like to start with this one. So microcosm experiments and computer simulations that I've been using, I think they are really powerful in telling us what's plausible, what may be plausible in nature. But of course, these are just tools, and we of course want to know what's happening in nature. So it's really important, I think, to try to test these kind of ideas under field conditions. So that's what I was really interested in trying to get at. And I got really lucky in trying to do this because this is really coincidence, but I was talking to different people from the Netherlands. They had a really nice experiment, grassland experiment where they had these experimental plots. And this is really a nice experiment because from the perspective of immigration history, because what they did was they directly manipulate initial immigration history by sowing different plants into different plots. And after that, that was done in 1996, almost more than 10 years ago now. And since then, they've been monitoring community dynamics for a very long time now. And so this setting, although it's been designed for a slightly different purpose, this was actually a really good thing to try to look at the effect of immigration history under field conditions. So this is from Google Maps, and you can actually see these plots in the Netherlands, central Netherlands. And if you see these plots, what they did was they had a block design, and each of these, what they did was they saw different species initially. This was a bare ground, and they saw different set of species of plants in 1996. And to have replicas, they repeated that for 5 times. And they were looking at how similar these communities, initially different communities, would become, or how dissimilar they would become over time. So looking at species composition, I'm going to look at community dissimilarity or how different they are. And this is where they did initial species composition manipulation. And 1 year after that, this is where they are. This is only relative, but this is very different communities. So this is really comparing 3 different communities where different species was on. And theoretically speaking, conceptually speaking, what you would expect is that if this dissimilarity decrease over time, that would mean that these communities are becoming more and more similar. So what initial difference you experimentally had is going to disappear. So if you have this kind of convergence, then you would conclude that immigration history is not important. On the other hand, if you have divergence with communities becoming more and more dissimilar, or at least no convergence, then you would say that immigration history is important to try to explain what's happening here. You have to go back to what happened here to explain community structure here. And this is the data, and it's really, if you do a statistical analysis, it's really no convergence. And if you look at, this is the photograph from some of the plots. After, this is almost 9 years. And after 9 years, it's surprisingly different. And you can see it graphically. So immigration history seems like it is important. But you can do something more interesting with, with their data. So what we decided to do is to, instead of just looking at species composition, we made use of all the different information about plant traits to use that information to look at trait-based functional composition instead of species composition. And what we found is that in contrast to that, we did have statistically significant convergence. So it seems like it really depends on what aspects of community structure you want to understand. If you're interested in looking at exact species composition, then your answer is that immigration history is important. But if you are mainly interested in functional groups, functional composition, then immigration history wouldn't be important. And this kind of idea actually is not, I don't claim this idea to be new, but what's new here is that this is a really nice, I think to me, a neat experimental evidence that's so rare. And so we have this contrast between the two, depending on the level of organization you look at. And this first direction that I was taking kind of led to a second direction I became interested in. So if functional group composition is not sensitive to immigration history, what's the implication for that to the functioning communities as ecosystem? So that's the second question I've been trying to understand. How, if these two are going to affect community structure, does it going to also affect ecosystem processes driven by species here? If the functional composition is not sensitive to immigration history, then you might expect that this relationship is not there, because functionally if these are not sensitive to history, then this relationship wouldn't be there. But of course, this grassland experiment is only one of the things that you can do, and it may be that it really depends on ecological context, what context you construct your communities, and that might influence how strongly this community structure may affect ecosystem processes. So that's what I wanted to do with people, really nice people in New Zealand, and I have a collaboration there, ongoing one, to look at wood decay fungi as a model system. And it's really a nice model system, I think, to do this, because they are known to compete strongly over this resource wood locally, and there's a lot of species, particularly in this New Zealand beech forest, a lot of wood decay fungi. So it's a diverse system that both taxonomically and functionally, so we can look at how these species assembly may affect ecosystem processes. And of course, needless to say, we kind of tend to forget about these things, but of course, without fungi, wood decomposition wouldn't happen. So obviously, this is a really key player for important ecosystem processes in forest ecosystem. So these are the collaborators I've been working with. So I don't claim I'm a mycologist, so I have to really work with different people. So mycologists, forest ecologists, and molecular ecologists, we've been working together to do experiment, both in the lab and the field. I'm going to talk about a field lab experiment, but I wanted to emphasize that we're also trying to do, and we just started field experiments as well. But here in the lab, what we do is we have this kind of small wood disk sterilized, and we have 10 species here. And to manipulate immigration history, we had 10 species, and one of them, we put them first, and we waited several weeks until we put all 9 others there. So in this case, this was, I think, the first species. So they are spreading by the time we put these ones. And we're also interested in how immigration history may interact with nutrient levels, just like this kind of similar way for the productivity diversity relationship question that I was trying to answer. And we have 5 replicates per treatment. So this is really a kind of a still incomplete analysis, but nonetheless, I wanted to show you these results. Really, we just got this data. So looking at fungal community structure, we are seeing a huge effect of immigration history on fungal community composition. So the way you see this graph is that these are the first species introduced, and we can look at different species, and I only show 3 species. And if you look at this species is really competitive. So they are really dominant, except in one case. When this species first introduced, they don't do well. So there's a huge effect here. And if you look at this species, then this one is doing really well, but not when they are not first introduced or first species introduced. And similar thing happens here. For this species, when they are introduced first, they do really well and become dominant, but they are not as well. They don't do as well otherwise. But the how well they do also depend on which species were introduced first. We're also having really encouraging results on ecosystem process component 2. So if you look at wood decomposition, how quickly wood is losing mass, and also the ratio of carbon and nitrogen within wood, which is important indicator of ecosystem processes. What we have is a clear effect of first species introduced, but not only that, if you look at initial nutrient level, which is another thing that we manipulated, we haven't really find a neat way to summarize this and generalize this, but it's at least clear that in both cases, there is some complicated interactive effects of initial nutrient level and first species introduced. Additional thing I wanted to show you quickly is we also trying to manipulate not only nutrient level, but also fungal consumption as an additional factor that might interact with immigration history. And we're using these little cute collembola, springtails, as fungal consumers, little tiny animals. And so I'm just going to show you a few pictures here, just representative photos. We just got this really just last month. And so you can see that there's a huge difference, and this is only initial stage, but nonetheless, you do have a big difference here. One species dominating here, but not here. But if you put fungal consumers, collembola, they really like this species, and it's almost gone, and it's going to allow other fungal species to potentially take over those species that may be resistant to this fungal consumption. But if you look at what's happening here, then although the collembola are around, they are too small that you may not be able to see it, but this one is not much different, and this is very different. So this is really still a very early stage of this experiment. But we are kind of seeing it's becoming likely that we do going to see some effect of interactive effect of consumption and immigration history as well. So these things, as example of ecological context, these things could interact to affect fungal community structure. And at least from this experiment, it seems like we do have a huge effect, flow-on effect from here, in turn, affecting decomposition, nutrient cycling. So that was the second direction I was trying to take with collaborators. And going to the third direction, I was getting really interested also in not only looking at ecological context, but also evolutionary context and how these two may interact to affect community structure. That's, I think, to me, really new, to my knowledge, I think it's really new, exciting direction, I think, too. And so for this one, I like to go back a little bit to theory of island biogeography. So we talked about immigration and extinction rate. And another way, separate from immigration history research, another line of research that other people have been getting really interested in is to try to combine evolution dynamics, evolution diversification, to say that it's not only, if you want to explain species diversity here, you shouldn't only look at immigration and extinction rate, but you have to combine three things together to see species diversity as a balance between these three things. And there are a lot of really interesting studies going on, particularly in using Hawaiian spiders and Caribbean Anolis lizards from the Caribbean, and these papers and many others, trying to combine these two, and that's a really new, I think, direction. But as I say, this line of research is kind of have been done more or less separately from this kind of relationship that I've been interested in from ecological time scale. So the basic, I was just curious to see whether it's possible whether this immigration history could also affect diversification, not just extinction over ecological time scale, but also diversification over evolutionary time scale. Now, if you want to do experiments on this, you quickly realize that's very difficult. If you want to do experiments on spiders or lizards or any other, most of the things, organisms, it's just not possible. Evolution takes a long time, 1,000 years or many more. But you really have to do experiments, I think, if you ask me, if you want to rigorously test the effect of history on anything. So that's a challenge, but there's one unique organism that can do this, and it's going back to microbial systems. And what I have done is to take advantage of this bacterium system where a lot of people have been using to look at adaptive radiation, to see, to use this as another model system. Just to give you a brief background, these people almost 10 years ago now show that if you put ancestral genotype into this small vial, what they found is that they quickly, so just like protists and even shorter time, generation time, they quickly saturate and they reach carrying capacity within a day or two, and they quickly reach carrying capacity. But after that, a few days later, something interesting happens. And I should say that we can consider this as a generalist because they go anywhere in this microcosm. But few days later, you get something interesting, so that through mutational processes, it's really genetic changes. They know that through mutation, you get new genotypes. And these are specialists in the sense that some of them use only surface, and this is surface specialists, and some of them use bottom specialists. And this is an example of adaptive radiation. And it's been also known that within this community, within this broad group, you can also have multiple species existing. So using this system, what I wanted to do is, going back to immigration history idea, I had 6 different immigration histories experimentally manipulated with 3 lipids per treatment. And I just used 2 genotypes to see what happens here. And both of them came from single genotypes, so that we can be sure that any new types are coming from within this through mutation. And another nice thing is that you can actually distinguish between these 2 because one of them is marked by neutral genetic marker. So quickly, what I'm going to show is the results over time, what was happening. If I introduce this genotype only, this is what I found. So there's a quick reach in carrying capacity, but after that there's a diversification. So if you just simply count the number of genotypes here, you see this diversification. If you put this one on day 4, they are growing, but they are really not affecting this diversification. So you compare to this one, you see a similar kind of diversification here. If you put this one a little earlier on day 1, they are growing really well. But compared to that, if you look at diversification, it's not really affecting. Although there is some effect on population dynamics, it's not really affecting overall diversification. But what I'm going to show you next, is going to be very different from here. So I put this one really early, just 6 hours after this introduction, and almost all diversification is suppressed. And that's the same here. This one is growing well, and this time it's giving rise to this type themselves. But because this one is similar, identical to this, it doesn't really contribute to diversity. And the same thing happens here. If you put this one in this immigration timing, you don't see diversification. And it's the same here. If you put this one here, then there's no diversification coming from this from this one. So the obvious question is, how did history affect diversification? And for the fuzzy splitter, this is another name we use for this one. It's been known that there are some competitive interactions so that this one can invade this generalist, but this one can't invade this one because of competitive interaction, mainly for oxygen and nutrients. And it's been also known that these two compete with each other. So if you have this kind of prior knowledge, you can think of this immigration history. And if you have this one introduced first and then this one, then what happened is that this one is going to inhibit this one. So this one is not abundant. And because of that, this blue one is indirectly facilitating the emergence of this green one to invade this one. But if you have the opposite one, then this red one is going to be very abundant. And because of that, although this one itself can be there, because this is too abundant, this is suppressed. So it's really indirect facilitation depends on history. And for these species, I don't have time to show you the data, but it's, I just show you, say that the secret of this historical effects was really a competitive equivalence. These are competitively equivalent, and that the history becomes really sensitive. So using the same kind of conceptual framework, these two can also interact with each other to result in variety of patterns between evolution time and diversification dynamics. Another, the last thing that I wanted to talk to you about briefly is about more of an applied issue. Instead of looking at immigration history, I was also becoming interested in the extinction history, more of a species removal history by humans and how that might affect community structure. In this case, from the applied perspective, how that might affect, if you want to restore native communities, how might affect restoration of community structure, and how that might depend on contexts like habitat size and connectivity. And for this one, I haven't studied this one yet, but we a lot of people in Hawaii, as collaboration, we are talking about using this forest fragments created by lava flow called Kipuka in Hawaiian words, in this big island. And this is a really nice system because you have many of them isolated within lava flow. And what we are planning to do is to remove different kind of mammals from in different sequences and timing to see whether that kind of experimental removal is going to affect native species recovery, not only birds, but also vegetation and arthropods, and how that might going to affect community reassembly after disassembly. So to summarize, I started from this immigration history. We knew that this could theoretically affect community structure, but we weren't so clear under what conditions this relationship is going to be strong or weak. And I was trying to make this a little bit bigger picture so that we have more of an integrative historical perspective on ecological community assembly. And I'm becoming more interested in both basic and applied questions, and I would like to continue more of a microbial and computer simulation work as a kind of a tool to generate ideas, but I also like to also do field work with collaborators. Thank you very much.
Let me ask you the first question, if I might. In connection with the role of the historical, or of the immigration history, a number of us have done simulations of how the connectivity of the species influences the likelihood that some of the species are keystone species. So it's like your last point involving the disassembly of the species, the disassembly of the community. And it was pretty clear that if the community matrix had a lot of entries, that once the community built up to a large level, a lot of the species were keystones. And if you picked any one out, you'd have a cascade of subsequent extinctions. So it would be very history dependent as to what would happen. So the bottom line was that the extent of the interactions in the community matrix controlled the sensitivity of the community to historical effects. And I'm wondering if you can pick that up here, if you can find sparse or interactions which don't extend across a lot of species, but are just sort of pairwise, stuck being pairwise. If you get very little influence of history in those communities, but meanwhile, if you have other communities in which every species is interacting with everybody else, they're the ones in which all hell breaks loose, depending on the colonization history.
Yeah, I think that's a really interesting idea. And from the work that I showed you, I haven't really gotten into the details of the connectance, how species are connected, just like the way you describe it. So I think one good direction is to really look more in detail what food web structure that these communities that assembled look like, and see and removing species. That's a good way to look at. It's really about, if I understand your question right, it's really about higher order properties of communities. So we are really thinking about historical effects on the community stability. And I think it will be interesting to remove species and see whether how robust the communities are to species removal may affect immigration history too. Yeah.
In the last series of experiments, two things. Does it matter which two species you put in first close together, whether or not you can't make a third one invade? And
So you.
The second thing is if those two not species genotypes, if the two genotypes are there, are they utilizing different parts of the resource? And if you put the third one in a different part of the resource, will it not invade? And why do you think it is that you can't get a third species, a third genotype to come in?
So yeah, get into the second question first. So there are mainly three groups as I described. So one is generalist and one is surface specialist, and another one is bottom specialist. And it seems like the surface specialists and generalists seem to be able to coexist regardless of history. But it's really the third group, which is bottom specialist, that's really sensitive to the history of two other groups. And maybe I'm not really answering.
Why use that? If the bottom is still available, it seems that it should be able to use that resource.
Yeah, that's something that I didn't explain well. Yeah, the key here is oxygen availability. So oxygen, of course, comes from the top. And if you have a thick surface by the surface specialist, that's going to block oxygen coming into the microcosm. And so it's really a disadvantage if you are a bottom specialist, and if you have already a thick layer of surface specialist here, and that would inhibit bottom specialist. But if you have a generalist, then they have a competition with the surface specialist, so that as I said, generalists can indirectly facilitate bottom specialists because they can inhibit the development of thick layer, so that the oxygen is more available to bottom specialists.
Steve?
Yeah, I want to thank you once again for demonstrating why rocket science is easy and ecology is hard. I mean, the former may have complex equations, but there's generally deterministic systems that when you know the boundary conditions of the history, you get a unique equilibrium. Here, you're showing high context dependence, pathway dependence, and initial condition dependence. So my question is, that makes me pretty depressed thinking about somebody who wants to occasionally look at restoration ecology. If everything is context dependent, what kind of generalizable predictability will we have, which we want to do in order to try to be efficient in any sort of restoration activities? And I have a second related question is, in the modeling part we did with the Lockyer-Volterra equation, I mean, I thought the results were very interesting, but isn't everything in the interaction coefficients, and where did they come from?
Yeah, both really, yeah, good questions. And the first question, it is depressing, but at the same time intriguing. But to try to give you a more encouraging message, I can give you two things. One is that if you know under what conditions immigration history is more important and under what other conditions immigration history is not important, then it's a little bit one step level above, but you can at least try to predict under what conditions you would expect a historical contingency and what other conditions you would expect to have a more deterministic path. So that kind of information might be useful. And the second thing I can try to say is that it's kind of related to the species pool simulation. It also could depend on scale, spatial scale. So these things that I showed you, it seemed like history is very important, and that could be discouraging, but it may be at some cases only happens at really small spatial scale. So if you look at large scale, maybe things is more encouraging and more deterministic. So that spatial scale is another thing I didn't talk about here, but I think spatial scale is another key that we should look into more to think about historical effects. And going to the second question you had, Lockyer-Volterra equation, it is true that it's very sensitive to the interaction metrics. And what I did here was that I was following some other people's method of trying to define interaction strengths based on body size, predator-prey interactions. There's some empirical relationship between relative body size between predator and prey, and how strongly they interact. And based on that, you can come up with, it's far from perfect, but it's, I think, better compared to random assignment of interaction metrics. So, so that's what I did, and hopefully it's a little bit more closer to reality.
In some of your examples, you looked explicitly at functional diversity and sometimes sort of implicitly at functional diversity. And I guess I'm wondering whether you think that there's more likelihood of a predictability or deterministic outcome when you think about functional groupings, and how do you decide what those functional groupings should be as opposed to, say, a species composition?
Yeah, yeah, that's a good point too. Yeah, I do agree that I think functional composition overall tend to be more deterministic and more predictable than specific species composition. But in response to the question, well, how you define what would be the good way to define functional groups? That's whole big area of research, and I think it's really specific to systems, so I don't have a good answer to that. But it really, what's important functionally really depends on what your interest is. And from the perspective of ecological restoration, if you want to know the productivity, you look at traits that are important to that, obviously. Yeah. But in conservation, we do also want to conserve particular species, and if that's also a big conservation goal, then it's really challenging because species composition can be really sensitive to history.
So in the Process experiment, the first experiment, you just label immigration histories, just ABC, so we don't know what they are, but you obviously do. So do you have a sense of, is there a rhyme or reason to what kind of immigration history leads to different relationships? Any insight other than anything can go any which way?
I don't have a good idea. What I was doing is I was randomly assigned, yeah, so that's kind of a simulation approach. I was randomly assigning different species into different histories. So it's kind of, I think, related to John's questions that we really have to look at full web structure to get to the next stage, to be able to have predictive power of in relation to historical effects. So I don't have a good answer to your question.
Great, Gretchen.
So yeah, just building on these questions, I was curious about the plots you study in the Netherlands. What's your thinking in terms of the amount of time you need to show the probability of convergence? It seemed that I couldn't tell exactly what was growing there, but that 9 years might not be sufficient time to expect any real change. That you might, I mean, those weren't all annual plants, right?
Some of them are perennial.
Some are perennial. So I just wondered where you're heading with that experiment, and I guess I imagined when you presented it that maybe it would take much longer in terms of generation time to see any clear answer to your question.
Yes.
The role of history.
Yes. Yeah, that's a good point. 10 years is a long time compared to a month that I use for microbial experiment. But relatively speaking, month is really long time or longer. So here, I shouldn't say that they are going to, they have reached some alternative stable state. I should never claim that these plant communities have reached some stable state and they never converge. So I think your point is well taken that the given longer time, species composition too may eventually converge. But I think sometimes we put too much emphasis on stable states, and I think the transient dynamics is also interesting and important. So the difference between functional composition and species composition, it's still there after 9 years. So that difference, I think, is significant enough. But I think it's possible that the species composition is also going to converge eventually. And good news is that it's really impressive work here, that they are still continuing this work. So they are monitoring this community even now. So we may be able to answer that sometime.
Okay, Mark, last question.
In the Pseudomonas experiments, is there any way that you can see whether the presence of a phage would change the result of that experiment?
Some other people have already been trying to introduce bacteriophage, and in relation to historical effects, I haven't done anything, but that would definitely be an interesting thing to do, and that's a little bit related to fungal composition. So we know that predation can be an important factor determining historical effects. So I think it will partly depend on how specific the phage is in attacking these different genotypes. If they are really generalist, it may not matter. But if they have some preference in attacking some of these genotypes, that could lead to some amplification of historical effects. But that's as much as I could say now.
I was thinking more in terms of the possibility of gene transfer from one of the levels to another, which might shift everything.
Okay, yeah, that's a new thing to me, and yeah, I should look into that too.
Okay, you have a long ride home.
Thank you. Thank you.